Okay, guys. So, next is the cell structure, function, and transport unit. This was unit two. This was when we were talking about prokaryotic and eukaryotic cells and plant and animal cells, cell theory, microscopes, all that fun stuff. So, these are the performance expectations. I know it looks like a lot, but believe it or not, you guys already know this stuff. So let's just get right into it. So students will compare, contrast these types of microscopes. So we've got a compound, a dissecting, a scanning, and a transmission. And then personally don't agree with remembering the names of dead dudes, but whatever. That's up to the county. So I said I have it all prepared in these flashcards I made for you guys. So the flashcards for this unit is biology, cells, and microscopes. So just getting right into it, the microscopes that you guys are used to using, it's called a compound light microscope. It uses one lens to magnify an object. Okay, so it's like we're taking a bunch of magnifying glass and just increasing and increasing and increasing the view till we can get a good picture of what we're looking at. So this thing that you're looking at right here, the ones that you see in the classroom, these are compound light microscopes. This one, a dissecting microscope, we don't use these very often because they're very low magnification. Mostly just what they want you to know is that the dissecting microscope uses the reflected light to form a virtual image instead of light passing through. In this one, light is going through the specimen. Light is going through the specimen in the compound. In a dissecting, it's just forming a virtual image. It's very low magnification. We usually don't use these. It's two you're typically only going to see at universities. You guys take higher level science courses. Actually pretty easy to understand the difference between these two. Because when you think scanning, you think, you're scanning the barcode, you're looking at the surface of something. When you think, transmission, something's going through. So a scanning electron microscope uses electron beams to scan the surface of a sample.
Microscope uses an electron beam to scan the surface.
No. Okay. So it uses electron beams to scan the surface. Okay. That's the critical difference between a scanning electron microscope and a transmission electron microscope. Okay. Scanning is just looking at the surface. The transmission, looking through and through. It's kind of like the difference between the compound and the dissecting. The compound is looking through it. The dissecting is looking at the surface. The only difference is these two use light. These two use electrons. I know it can get confusing the first time you look at it, but conveniently, this is not the first time you've looked at this. I would just take another look at the flashcards, maybe watch a couple YouTube videos. These things are universal. If you just Google these things, they'll show up. So that's all you need to do for those. Understand they want you to know these types of microscopes. And then for this, just gave you a very just brief overview. Hope they don't ask too many questions about these guys. I really don't like it when they expect you to know the names of scientists because it's whatever. But all right, so Robert Hooke was the one who discovered cells. If you need to remember it with a mnemonic or something, just remember that this guy got us hooked on the idea of cells. Haha. All right, so Robert Hooke discovered cells and got hooked. Okay, cells, the whole shebang. Bacteria was Van Leeuwenhoek. You can remember that as bacteria are dirty. And whenever I say Van Leeuwenhoek, it feels dirty in my mouth because, it's not a, I don't know, man. Listen, I'm trying my best here. All right, Van Leeuwenhoek. It's just hard to say and it makes my mouth feel gross. So he discovered bacteria. Schleiden concluded that all plants are made of cells. So he was just sliding in the fact that plants are made of cells, those things that book got us hooked on. Animals concluded that animals are made of cells. Schwann, because a swan is an animal and a swan is made of cells. So Schwann concluded that animals are made of cells. Chow for chow, whatever, determined that all cells come from pre-existing cells. So at first we started with one cell and then we were like, for cow, no, we got two. I don't know. Okay. Listen, do whatever you need to do to remember it. Yeah. So Virchow or Virchow concluded that all cells come from pre-existing cells. Schwann was like the animal. Animals are made of cells. Schleiden was just Schleiden in the fact that plants are made of cells. Then Leavenworth discovered bacteria. Robert Hooke discovered cells. Modern cell theory states what these guys were talking about. All organisms are made of cells. So that was Schleiden and Schwann. That were like all organisms. Cells arise from pre-existing cells, and cells are the basic unit of all organisms. All right, that is modern cell theory. Some stupid people used to believe that life could just arise out of nothing because, they would look at a piece of rotten meat and go, oh, well, maggots came out of the meat, so the maggots just showed up out of nowhere. And then a bunch of scientists had to go, no, you're a moron. Flies left the eggs in for the maggots, but that's, whole other set of experiments. So going back, so just know the differences between these microscopes, know about these scientists guys, modern cell theory. Okay. And then they also want you to know scientific theory versus scientific law. So theory. So for example, a lot of people misuse the word theory. Some people use it to mean they just have a guess. No. A theory is a well-tested explanation. So for example, gravity is a theory. All right. And we all know that when you jump up, unless some crazy stuff is happening, you are going to come down. All right. So that's what that means. Scientific law, a precise statement that summarizes the results of many observations and experiments. So a law versus a theory. Laws tend to describe a narrower set of conditions. It might explain the relationship between two specific forces or between two changing substances in the chemical reaction. Theories are more expansive and they focus on the why or how. So, you might see the law of motion. They stay in motion until acted upon by an outside force. That's a very specific thing. Gravity is just like, hey, big things, pull things in. Ain't that neat? You won't have to worry. Now, I hope that they don't test you guys too much on these first bits in this. If I had to guess, there's probably only going to be a total of maybe 3 questions from all of these things, maybe 3 to 5, if that. Now, the real thing that they're really going to be emphasizing is what we go on from here. So we went over it for a while, prokaryotic and eukaryotic cells. All right. They have some similarities, but they also have a lot of differences. So a prokaryote, okay, it does not have a membrane enclosed nucleus. So a prokaryotic cell, these are very simple cells. Okay. These are not what are composing the living things that we think of. So like animals, plants, things we think of, those are all eukaryotic. Prokaryotic cells are stuff like, bacteria and little nasty things like that. So a prokaryotic cell does not have a nucleus and it does not have membrane-bound organelles. What that means is that imagine if your heart and lungs were not fixed in a set spot in your body and they were just floating around inside of you. That's kind of what's happening in prokaryotic cells. Their organs are not set in a specific location. So when we say does not have membrane-bound organelles, we mean that their organelles are just sort of in free run. Additionally, you guys remember where DNA is stored in a eukaryotic cell? The nucleus, that's right. So because prokaryotic cells do not have a nucleus, they store their DNA as plasmids. So plasmids are those round things. It's DNA that's stored as kind of like just a round circular structure. So these things are plasmids, okay? That's how prokaryotes store their DNA. We do not store our DNA like this. No, this is for simpletons. Okay. We are complex beings. We need something with a bit more structure. All right. So those are the critical differences between prokaryotic and eukaryotic cells. Prokaryotic cells have their DNA stored as plasmids. Prokaryotic cells do not have a nucleus. Our organelles are not membrane bound. And the two types of prokaryotes are bacteria and archaea. All right. Then eukaryotic cells, these are the things that make up us. We are made of eukaryotic cells. Your dog is made of eukaryotic cells. Your plants are made of eukaryotic cells. All that's made of eukaryotic cells. So we have a nucleus. Our DNA is stored in the nuclei, and our organelles in our cells are bound by membranes. Okay. In terms of what the two have in common, they both have DNA. Granted, it's stored a little differently. DNA in a prokaryote is stored in those circular plasmids. DNA in a eukaryote is stored in the nucleus. Both have ribosomes because, everybody's got to make proteins. So we both have ribosomes. We both have a plasma membrane because remember, the plasma membrane is just that outer shell covering. It's like the skin of a cell. Cytoplasm, so that jelly stuff that's filling up the whole thing. So that is something they all have in common. And you guys need to remember this because I guarantee it's going to be at least 1 to 2 questions. Ribosomes, plasma membrane, cytoplasm, DNA. Okay. Just the basic things that a living thing needs to live. Okay. You need ribosomes because you need to make proteins. You need a plasma membrane because imagine living without your skin. You need a cytoplasm because, it's not just free air floating around. You need that jelly to sort of suspend everything. It needs DNA because you can't live without DNA. It's what makes you, you. It's the building block. It's the blueprint. Now, plant and animal cells. So these are a type of eukaryotic cells, plant and animal cells. Plant cells have a cell wall and chloroplast, but animal cells do not. All right. A chloroplast is what's letting the plant cell do photosynthesis. It's letting the sunlight in so that it can convert it into sugars for energy. Okay. Also, just you don't need to sit through all this at once. The second you feel yourself spacing out during any of these videos, pause, chill for a second. I'm not going anywhere. All right. But yeah, so plants have a cell wall and chloroplast, but animal cells do not. Okay. Animal cells do not have a cell wall. Cell wall is a very rigid structure that helps give plants sort of their general shape. It allows them to hold themselves up. We are held up by specific structures. We've got bones that help, hold us upright and muscles to do all that. Plants don't have muscles or bones. So their individual cells need those cell walls to give them a more sturdy structure. All right. So plants have a cell wall and chloroplast, but we animal cells do not. All right. So animal cells and plant cells have in common. We have in common anything that makes a eukaryote a eukaryote. So we both have a nucleus. We both have membrane-bound organelles. We both have a mitochondria. All right. So, we both just have everything that a eukaryote is characterized by. Plant and animal cells have it. All right. The plasma membrane is also called the cell membrane. It's that outer layer of the cell. It regulates what enters and leaves. It's called selective permeability. Selective permeability. What permeability means is the ability to permit something when you break it down. So it selectively allows what comes in and what leaves the cell. All right. It's kind of like the bouncer at a club. He's not just letting anybody in. All right. You gotta have permission. All right. So the cell is only letting, things that it wants inside. So like nutrients and stuff like that. It typically doesn't allow stuff in that could potentially hurt it. All right. A nucleus. We've talked about what the nucleus is. It contains the DNA and the RNA responsible for growth and reproduction. All right. So the nucleus, that's where we have all of our information stored. Within the nucleus is a specialized structure called the nucleolus. That's where ribosomes are made. A sneak peek into the fifth unit. Remember, ribosomes are RNA, ribosomal RNA. So that's why we make it in the nucleolus, because, how do we get RNA? From complementary base pairs matching DNA. But that's from, well, we'll get into that more in the fifth unit. For now, I just want you to know that the nucleolus is where we make our ribosomes. All right. A rough endoplasmic reticulum. I just want you to know that it's this stuff right here. You see the stuff with little dots on it surrounding the nucleus. This is where we make our proteins. And the reason it's rough is because it's lined with ribosomes. And you remember what ribosomes do, right? We'll have a Dora the Explorer moment. What do ribosomes do? Make proteins based on the sequence of messenger RNA. Very good. So that's why this is where we're making all these proteins, because the rough endoplasmic reticulum is lined with ribosomes, all right? So that's what we mean when we're talking about the rough endoplasmic reticulum. Then the smooth endoplasmic reticulum. This is the part that does not have all those ribosomes attached to it. This is more involved in the synthesis and secretion of lipids, all right? So the rough ER, making our proteins. Smooth ER, secreting our lipids. They do want you to know as well the relationship between the rough ER, the Golgi apparatus, and vesicles. Or I'm sorry, vacuoles. I'm sorry, vesicles, vacuole. All right, you know what? We'll restart. Anyway, so the rough ER makes the proteins. The Golgi apparatus is sort of like the Amazon, they're packing and shipping them out. All right. Store in the vesicles. We use vesicles to just sort of move stuff around. They're kind of like just bubbles that are holding whatever we need until they get where they need to go. Vacuoles, not that common in animal cells. It's more of a plant cell thing. It's sort of just like a warehouse where they store everything. We already talked about ribosomes. Those are what make the proteins. All right. So, this is all stuff you guys are pretty familiar with. Yeah, we got some scientists. Yeah, we got some microscopes, but they're probably not going to emphasize that too much. You need to know modern cell theory, though. You need to know all organisms composed of cells. They come from pre-existing cells at the basic unit. All right. You need to know the differences between prokaryotic and eukaryotic cells. You need to know what they have in common. You need to know these organelles. Ribosomes make proteins. Osmosis is the diffusion of water through a selectively permeable membrane, okay? So water will move on its own through a selectively permeable membrane in order to balance out the concentration, all right? Then we're going to talk about passive and active transport. So active transport, it's when it's moving against the concentration difference. So remember, this is with almost everything in science. An area of high concentration will always naturally want to move to an area of low concentration. For example, you're squeezing a tube of toothpaste. The reason that toothpaste comes out of the tube is because you're applying pressure to the tube. So the toothpaste wants to go from an area of high pressure to an area of low pressure. It's the same thing with water and anything else. There's a high water concentration over here, so it wants to move to an area with lower water concentration. All right, because due to not as many sugars over here, the ratio of sugar to water is high, so water wants to move that way. So that's why I want you to know water moving through osmosis is passive transport. It's passive because it requires no energy. All right. Passive transport is when we're going from high to low concentration. All right. Again, it requires no energy. It's passive. It requires no energy. We're going from an area of high concentration to an area of low concentration. Active transport is the opposite. If we want to go from an area of low concentration to an area of high concentration, we have to move against the gradient, and that requires energy. We're going against so for example, let's say you're in a current in a river, okay? Which direction are you going to go if you don't do anything at all? You're going to go with the flow of the river. Now, what if you wanted to go opposite the direction of the river? You're going to have to use a lot of energy, right? You're going to have to swim pretty hard. Not a lot of us can do that. So that would be active because you're going against the gradient. Okay. So you need to know that osmosis is a passive process. Okay. Because water is moving from an area of high water concentration to an area of low water concentration. That's osmosis. Next, we're just going to get into a couple more structures. So cilia, all right, just these tiny little hairs, that's cilia. Flagella is a more specific singular structure, like a singular tail. All right, that's flagella. Flagella is more for a specific direction. Cilia are just hairs kind of all over the place. All right, I know these organelles can be a lot to keep track of, but again, just look at the flashcards, listen to these videos, do whatever you need to do. All right, it won't take you that long to get it done, I promise. Lysosome is a organelle filled with enzymes needed to break down certain materials in the cell. So think of this as kind of like the stomach. It's breaking down certain things within the cell and coming up with the waste product. All right, so that's what our lysosome does. Whenever you see the prefix LYS, that usually is meaning we're breaking something down within the cell. All right. Then we've got the mitochondria. It's the powerhouse of the cell. You've all seen the memes. Okay. It's where we make all of our energy. All right. We've already talked about a chloroplast. This is where photosynthesis occurs. So this is where the magic happens. Sunlight comes in, they make some sugars. We've already talked about selective permeability. It allows some substances to cross the membrane more easily than others. It's selective about what comes in and what goes out. That's why it's called selective permeability. We've already talked about diffusion. It's a movement of molecules from an area of higher concentration to an area of lower concentration. We're going to talk about tonic solutions in a different video because that can be very difficult to grasp. So I'm going to address that in a video all on its own. And then just the parts of a ribosome. We got the large subunit, which allows the transfer RNA to bind. And we've got the small subunit that's reading the messenger RNA. So the small subunit is what's reading it. A large subunit is what's bringing in the amino acids. So here, let me see if I can find the basic picture of a ribosome. So here we go. So here, the small subunit is what's reading this line. The large subunit is what's bringing in what we need to make our proteins. So that's pretty much just a quick rundown of your second unit. I know that was a lot of information. Thankfully, though, you guys have already gone over it. So yeah, we've already talked about all that. All right. And that's pretty much just unit 2. Like I said, just make sure you're just looking at your flashcards, watch the video once or twice, whatever you need to do. You don't need to stress out, spend 8 hours a day studying. No, just like I've been saying, just 10 to 15 minutes a day. That's it. Okay. I don't give you guys homework, just 10 to 15 minutes a day and you'll be Gucci guilty. Okay. Yeah, this will look pretty good. And I think next we're doing mitosis and meiosis, and then we'll be doing the stuff that we're doing at the end of the 5th and 6th units. But yeah, I'm very good at ending videos. I can't tell. See ya.